Okay, hi everyone. So today I will be continuing on our lecture. For the last lecture, I've stopped until auxochrome. Okay, so now I'll be continuing on explaining what is auxochrome. So auxochrome is basically a group of atoms that is attached to a chromophore. Means that you need to understand that a chromophore is a compound that can absorb UVB radiation, and an auxochrome is a group of atoms that is attached to an auxochrome, to a chromophore. Therefore, this attachment of auxochrome to a chromophore will modify the ability of the chromophore to absorb light. Okay, so this group of atoms, or we can call it a functional groups, does not absorb does not absorb in a UV region, but it has an effect on the absorption peaks where it will appear where it has an effect of shifting the chromophore peaks to a longer wavelength as well as increasing their intensity. Okay, so basically when we look at in terms of the spectrum, if it is just a chromophore without auxochrome, so maybe it will just be like this. So maybe the optimum one is at around 200 nanometer. So this is without auxochrome. So if it is a with an auxochrome, so you should expect a shift of the radiation of the wavelength, and then you should expect shift, and then you should expect increase intensity. Okay, so basically it will look like this, something like that. So it will increase intensity, maybe 300 nanometer increase intensity, and also uh, uh, increase or maybe shift to a much more longer wavelength. Okay, so basically that is an auxochrome, the function of auxochrome, and also what does an auxochrome has an effect to a chromophore. Okay, so this is basically an examples of what is auxochrome and what kind of wavelength that it corresponds to, right? So this is one of the question. So do you think cyclohexane can absorb UV radiation as strong as benzene? So can a cyclohexane absorb UV as a benzene? Can it do it? Okay, so you need to think. If yes, why? If no, why? Okay, so as usual, jot down on a piece of paper and upload it lah. Okay, right. So yeah, so this is an example of cyclohexane, and then this is a benzene. So you need to justify why cyclohexane or why benzene, if it can absorb or cannot absorb UV radiation. Okay. So now, so we'll be coming into the second part of the chapter, which is the UV spectrometry. Okay, so now this is the first instrument that you will learn throughout this semester. So the first instrument is UV spectrometry. Okay, say here. All right. So in all of the instrumentation, I've shown you before the schematic diagram, isn't it? So this is an example also of a diagram, of a schematic diagram, where for UV vis radiation, UV vis instrumentation, you would you would have sources. The first one, sources. The second one is a wavelength selector. The third one is a sample cell, or we can call it cuvette. The fourth one is a detector, and the fifth one is a signal processor, which is a readout device. Okay, so we will be looking into each of the instrumentation inside a UV-Vis spectrometer. Okay, so the radiation source must be a stable source of radiation energy. So this is the condition. The radiation source must be stable, and also we need to have a wavelength selector, which is in this case we are going to look at monochromator and also some filters. Okay, the third one is a sample cell, which is a transparent sample container. The fourth one is a detector, and the fifth one is the recorder, as what I've mentioned before. All right, so UV-Vis instruments, UV-Vis spectrometry is divided into three types, which is the first one is a single beam, the second one is a double beam spectrometer, and the third one is a multi-channel spectrometer. Okay, so you will be able to look at into different kind of UV-Vis instrumentation and what is the difference, how does the difference differs from each other. Okay, all right. So the first instrument, the first thing that you're going to look at is the instrument architectures of a single beam versus double beam. Okay, so this is a single beam UV-Vis radiation, UV-Vis spectrometry, sorry, and the second one is the double beam spectrometry. So what is the difference? Okay, so basically, uh, we are going to look at a schematic diagram. So when I'm talking about schematic diagram, this is this also can represent a schematic diagram. Okay, all right. So for both single beam and double beam UV spectrometry, it comes from a single source. Okay, a single source, but throughout the process, it has some difference between them. Okay, let's look into the single beam first. So it comes from the one source. So this source, this light source, will provide electromagnetic radiation to a filter, or we can call a monochromator, which is this is a wavelength selector lah. Okay. All right. So this wavelength selector then will select a particular wavelength that that you have intended. So that particular wavelength will be absorbed onto your reference cell, which contain your sample. So this is where you put your cuvette. Okay. And then the transmittance from the reference cell will be detected by a photo detector and then read out. Okay, so basically, for a single beam, you need to do your reference cell as well as your sample cell at a different time. So basically, you need to do your reference cell first, which is the measurement of a reference cell is for you to know the background noise, the background of the instrument. So then, whenever that you put your sample cell in, that sample cell spectrum deducted by the reference cell spectrum, then you will got your the corrected sample spectrum. Okay, so basically we need the reference cell as well as the sample cell to provide the correct sample spectrum for your analyte. So in this case, for a single beam, you need to measure it differently at a different time. You need to measure the reference first, and then you put your sample in, and then and then automatically it will deduct the reference cell, and then you got your corrected readout afterwards. But in a double beam, you would see that from a single source, it flows into the monochromator. Here you select your intended wavelength. So that intended wavelength will be divided into two. Where you can see that the measurement of your reference cell and the measurement of your sample cell is done simultaneously and also being detected simultaneously, and then it is straight away giving you the readout. So basically, in terms of the architectures of a single beam and a double beam, it has a major difference where uh, it splits for double beam, but for a single beam, it does not split the intended wavelength, so you need to measure it twice. But for a double beam, you need to measure it just once. Okay, so don't ever make a mistake of saying that double beam spectrometry have a two source, which is no. It comes from the just one source, and it's been divided into two. sources. Okay, so that is for difference between a single beam and a double beam. Okay, so types of UV instruments for a single beam. So this is what I've explained before. So it employs a single source of supply radiation to the sample, and then the background intern here. The keyword is intern. Advantage is that a single set of components is required. So basically, the instrument is much more cheaper as compared to the double beam, which is much more expensive. The disadvantage is that the separation correction must be done for background spectrum due to the solvent matrix interferences, which will take much more longer time lah, because you need to do the measurement separately. Okay, so this is the same one. Okay, so for a double beam, as what I have mentioned before, it comes from a single source with equal intensity beams throughout the sample and the reference cell, which are then dispersed and being detected alternately. So basically, this beam splitter is important in terms of having it being measured simultaneously. Okay, right. So for a multi-channel spectrometer, I don't think that this is important. Sorry, I don't think that this is important for you. It's just that for you to be able to know on the difference between the single beam and the double beam spectrometer. Okay, right. So now we are going to look into each of the instrumentation part. Okay, so the first one is the sources. So sources tu macam mana? Sources ni is something that provides electromagnetic radiation. So we have a different kind of sources, which in this case kita panggil lamp lah. We can just call it as a lamp. So for the sources, you will have a deuterium lamp and a halogen lamp, which covers the UV range from 200 to 330 nanometer. If you are trying to look at much more wider range than that, you may use a tungsten lamp, which is corresponds to 330 to 700 nanometer. You can also have a xenon arc lamp, which corresponds to the same the same range of wavelength. So basically, what you need to know is that for the sources, it needs to be stable. Okay, it needs to be stable and it needs to have the correct range. So if you want to test the wavelength at, for example, 350 nanometer, you cannot use deuterium or halogen lamp. Okay, so for these slides, you can see that we have a lot of lamp here that corresponds to the wavelength. So basically, in this case, we have documented it for you to be able to choose whichever lamp that you deem suitable for your analytes. Okay, all right. So it's the same thing as well. All right, so the light sources, as I mentioned before, it needs to the ideal light sources needs to be stable. So for in this case, it is stable from 200 nanometer to 1000 nanometer. Okay, it needs to be stable throughout. We cannot have an alternate thing. Okay, so basically the source will need to be stable for the electromagnetic radiation to be stable for your sample or your analytes to be excited, okay, to be absorbed. All right. So the second one, we are going to look at a wavelength selector. Okay, wavelength selector, we have looked at before this, you have heard about monochromator. So monochromator is a part of a wavelength selector, which is a spectrophotometer. While you are also going to learn about photometer, which uses filters to select wavelength. Okay, there are two types of filters, which is interference filters and absorption filters. It can only detect a single wavelength at a time. as compared to spectrophotometer, which is monochromator. It allows to scan various wavelength. Okay, the input may be continuum or a line sources. All right. So the second exercise for this lecture is that I want you to use your own words. Okay, don't follow the notes. You use your own words, your understanding uh, in terms of differentiating between interference filter and absorption filter. Okay, use your own word as usual. Jot down in a piece of paper, in the same paper as you punya apa, as your exercise before this, which is the difference between cyclohexane and benzene. So jot down on the same paper as well, tak apa, it's okay. You can just upload in e-learning later on. Okay, so the difference between absorption and interference filter, right? So I will leave that to you in terms of differentiating between the filter. Okay, so this is the advantage and the disadvantages in terms of this filter. So when you say low cost, why? When you say simplicity, why? When you say, for example, the disadvantage of you having a filter is lesser versatility. So why? So all of this needs to have a proper justification when you say it. Okay, so just jot down onto that piece of paper, and then we'll go from there. Okay, all right. So I will continuing on the wavelength selector with the monochromator here. So a monochromator, it has many parts inside the monochromator. Okay, so the first one, we will have an entrance slit, and then we have a culminating lens, a dispersive device, a focusing lens, and an exit slit. So basically all of this you can read in terms of the definition, but basically what usually that we do is that the function of the monochromator is just a basic of choosing it scans throughout the whole range of wavelength, and it select the intended wavelength that you have intended to choose. Okay, so that's why this is the entrance slit from your sources. Okay, from your sources, so it goes in here, and then we have a culminating lens, and then we have prism, and then a focusing lens. So whatever that goes out to through your exit slit is the wavelength that you have selected. Okay, right. So the operation of a monochromator is that, okay, so a polychromatic radiation from more than one wavelength enters the monochromator through the entrance slit. So this is what I said before. Your sources would, because your sources have a range, kan? Your sources, for example, your sources have a 220 nanometer to 330 nanometer, the wavelength. So this is a range of 100 nanometers will enter your entrance slit, and then the beam will be culminated and then strikes the dispersing element at an angle. So the beam is split into its component wavelength by the grating or the prism. Okay, so basically when you look at here, it will enter from an entrance slit, and then when it goes to the prism, here is the beginning of your selectivity. Okay, the prism would focusing your intended wavelength towards your focusing lens, and then goes out from to the exit slit. Okay. Alright, by moving the dispersing element or the exit slit, radiation of only a particular wavelength leaves the monochromator through the exit slit. So this is the operation of how a monochromator works. Alright, so basically this is important, yeah? Okay. Alright, so this is a diagram illustrator on how does monochromator works. Okay. Right. So basically both monochromator or filter, which is in this case, it is a wavelength selector. The function of wavelength selector is does come from its name, which is to isolate the desired wavelength so that only the band of interest is detected and measured. Okay. All right. So basically, this is the filter versus the monochromator. Okay. The filter would provide a low resolution of wavelength selection, while a monochromator has a high resolution. So filter this suitable for quantitative work. Monochromator usually we will have both lah, qualitative and quantitative. The wavelength cannot be varied for filter, but for monochromator the wavelength can be varied. But of course, monochromator is a little bit expensive as compared to just filter. All right. All right. So this is the wavelength selector, the choices of wavelength selector. So this is just for you to, for you to know, just for your knowledge, okay, in terms of how are you going to use your wavelength selector and where is the wavelength selector is optimum at, okay, right. So now we are going to look at into the third instrumentation part, which is the sample cell. So basically the sample cell, ataupun we can call it cuvette. I have shown you before, kan? Alright, so it is nothing new. It's just that you would need to know in terms of the material that is made for the sample cell and the corresponds wavelength requirements. So if you are doing something that is more than 300 nanometer, that less than 300 nanometer, you need to pick your cuvette or your sample cell material wisely. Okay. Otherwise, some of the material might absorb, some of the material might not absorb. Okay. Sorry. Okay, something happened. Sorry for that. We just look at into single pane. Okay. Sample cell. Okay. So now I'm sure that you need to know on the sample cell material that is being made of. So this is the same as what you have looked into the monochromator ke, the light sources ke. So we do have the material of for your cuvette. Okay, right. So there are some precautions, okay, for your sample cell. So you need to keep your cuvette or your sample cell as clean as possible because your thumbprint from your fingers will absorb or scatter the light before it enters the cell. Right? So basically, this is very important for you to get an accurate reading of your sample absorption. So don't get your fingerprints on them because your fingerprints will create some kind of deviations. Right. Use the same sample cell during running the blank, standard, and sample. So this is important as well of why we try to use the same ones. The same, even though it is not the same one spoon, it is just the same material for it because it will have the same deviations if it have deviations lah. If it is, yeah, we are here being logical of if something if you only have two cuvettes, so just use the two, don't buy a new one because those two would have the same deviations throughout the experiment. Okay, if you do have that one and you have cleaned everything, okay, so you just use that one during running the blank, standard, the sample. All right. So the sample, the solvent, so this is the common solvent that is being used inside the UV spectrometry. So you also need to figure out and point out on the suitable solvents for your analytes. Okay, so if you are doing something at through maybe you are doing something at 190 nanometers, so or water will be the suitable solvent. Acetonitrile also suitable lah. Don't use acetone because acetone is out of the range. Okay. Okay. So then yes, lastly we are going to look at the detectors. Okay, detector. The detectors must have a high sensitivity, have a high signal to noise ratio, a constant response for the wavelengths, and then a fast response time. So if it is just a single wavelength selector, usually we are using diode and then photo multiplier tube. Okay. Now we are going to look at into the detectors. Okay, so basically, when we are looking at UV spectrometer, so usually these ones lah, okay, around this area, okay. All right. So the first detector that we are going to look at is the photomultiplier tube, which is it detects one wavelength at a time. So basically, you need to know on how a photomultiplier tube works. Okay, all right. So now I'm going to explain on how photomultiplier tube works. All right. So a photon, which is the the light that you have been detected from your from your sample cell, which we call it photons. Photons strike the cathode and electrons are emitted. Okay, so basically, whenever that the photon strike the cathode, okay, it will generate electrons. Okay, so these electrons are accelerated towards a series of diodes. So we have diode 1 here, second one, third one, fourth one, fifth one. All right. So once it hit the first diodes, it will produce more electrons. So as you can see here, let me erase this first. All right. So this is the incident radiation, the photon, entering your photomultiplier tube. When it enters your photomultiplier tube, it bumps to the first diode. So from the first diode, it creates two electrons. So these two electrons will become 4, 4 will become 8. So this every time it hits a diode, it will double. Okay, so this is how you increase your detection intensity. All right, by using a photo multiplier tube. Additional electrons are generated at each diode. Okay, so once it reached a diode or the first diode, it will increase its electrons number, and then it hit the second diode, the third diode, the fourth diode. So you imagine that you would have a multiple electrons inside your photo multiplier tube. So this amplified signal is finally collected and measured at the anode. This is how a photo multiplier tube works. Okay. So this is an explanation also on how photo multiplier tubes function. So I believe that you can read this on your own. Okay, but this is also important for you to be able to explain it later on. Okay, I don't know why it keeps on shutting down. Sorry for that. Okay, a little bit more on the. Okay, so basically this is also another illustrative figures on how are you going to describe a photo multiplier tube. Okay, so by this you need to know on the importance of it lah. Okay. So the second one is a photodiode array. So this measures several wavelengths at once. All right. So basically, the photodiode consists of two semiconductors, which is the N type and the P type. So basically, the lights promotes electron into the conducting band, then generates an electron hole pair. Okay, so this concentration of electron hole pairs directly proportional to the incident light. So basically, it will produce a voltage which is present, and the concentration of the light induced electron hole pairs determines the current through the semiconductor. Okay, so this is the on how photodiode arrays measures your wavelength, right? So so basically, what would you expected from a UV spectrum is that, first of all, a UV spectrum is a plot of absorbance versus wavelength. So don't mix this up, of a sample at a different concentration. So you would imagine that if you have increased concentration, you will increase the absorbance. Okay, so because as Beer's law indicates that whenever that you have a high concentration, then you have a high absorbance. Okay, all right. All right, I think that I can stop at here now for this topic. I will continue on the next topic.